Welcome back, everyone, to another deep dive. Today, we're going to be taking a journey into the fascinating world of cells. You know, those tiny building blocks that make up all living things. We had a listener write in asking for a really thorough summary of this whole chapter from a biology textbook called A Tour of the Cell.
It's amazing to think about, right? That something so small can be so complex.
Yeah, and to understand how cells work is like unlocking the secrets to life itself. So where should we even begin with something this complex?
Well, before we dive into all the inner workings of a cell, I think it's important to understand how scientists even study something we can't see with the naked eye.
Right, it's like trying to understand how a watch works without a magnifying glass.
Exactly. And that's where microscopes come in. The scientists have been using them for centuries to peer into this hidden world. The early light microscopes, like the ones used by Robert Hooke way back in the 1600s, were revolutionary, but they definitely had their limitations when it came to seeing those tiny organelles, those mini organs inside cells.
So how do we zoom in even further?
Well, the development of the electron microscope in the 1950s was a game changer. Instead of using light, electron microscopes use beams of electrons, which have a much shorter wavelength, allowing for way higher magnification and resolution. And that's how we get those incredible images of mitochondria, chloroplasts, and all those intricate cell structures.
I've seen those images, they're mind-blowing. Yeah, it's like exploring an alien landscape. But it's not just about magnification, right? There's also resolution and contrast.
You got it. Those are key factors in microscopy. Magnification is basically just how much bigger an image appears compared to the actual object. Resolution is like the ability to distinguish between two objects that are really close together. The finer the resolution, the clearer the details. And then contrast is the difference in brightness between different parts of the image. Basically, it makes those details stand out.
Okay, so it's like having a high-definition TV with great contrast. You get a much sharper, more vibrant picture.
Exactly. And microscopy techniques have just continued to advance over the years. We now have scanning electron microscopes, or SEMs, that give us these amazing 3D images of cell surfaces, and transmission electron microscopes, or TEMs, that let us see through thin sections of cells. It reveals their internal structure.
Wow, it's amazing how technology keeps pushing the boundaries of what we can see. I read about this newer technique called cryo-electron microscopy, or cryo-EM. I think it involves like freezing the samples or something.
You're right, cryo-EM has really revolutionized structural biology. It involves flash freezing samples to preserve them in their natural state, and then using an electron microscope to capture images. This technique has allowed scientists to figure out the structure of incredibly complex proteins and cellular machines in really fine detail. For example, ribosomes. Actually, the scientists who developed cryo-EM won a Nobel Prize in 2017 for their work.
Wow, impressive. But it's not just about visualizing these structures, is it? We also need to understand how those structures function, the chemistry behind it all.
Absolutely, and that's where biochemistry comes in. Biochemists study those chemical processes within cells, like how enzymes catalyze reactions, how energy is generated and used, and how molecules interact to carry out all of life's functions.
So it's like having this detailed map of a city and then figuring out like how the traffic flows, how the power grid works, how people interact.
That's a great analogy. One technique that combines microscopy and biochemistry is cell fractionation. It's kind of like taking a cell apart and separating all of its components.
Oh, like sorting laundry, but with cell parts.
Exactly. They use a centrifuge, a machine that spins samples at super high speed. This creates a force that separates components based on their size and density. It allows scientists to study individual organelles in bulk and figure out what they do. For example, that's how we know that mitochondria are the powerhouses of the cell. They're responsible for generating energy.
Okay, this is all incredibly cool, but let's zoom out for a second and talk big picture.
Yeah.
What are the basic building blocks of all living things?
Well, all living organisms are made up of cells, and there are two fundamental types, prokaryotic and eukaryotic. Prokaryotic cells, like bacteria and archaea, are a lot simpler and much smaller generally. They don't have a true nucleus or other membrane-bound organelles.
So they're like the studio apartments of the cell world.
That's a great way to put it. Now, eukaryotic cells, on the other hand, are more complex and larger, with a true nucleus containing their DNA and a bunch of membrane-bound organelles that carry out specialized functions.
So they're like the mansions of the cell world with
And
different rooms for different purposes.
exactly. And one of the key differences between these cell types is where their DNA is located. In eukaryotic cells, the DNA is enclosed within a membrane-bound nucleus, like a control center. But in prokaryotic cells, the DNA is just concentrated in a region called the nucleoid, which isn't enclosed by a membrane.
So it's more free-floating.
Yeah, you could say that. And that difference in structure really reflects the greater complexity of eukaryotic cells. Another key difference is the presence of those membrane-bound organelles. Eukaryotic cells have specialized compartments like the endoplasmic reticulum, Golgi apparatus, and mitochondria, each with its own specific function.
It's like a city with different departments all working together.
Precisely. Now, this difference in size and complexity is also related to something called the surface area to volume ratio. As a cell gets bigger, its volume increases faster than its surface area. This creates a challenge for nutrient uptake and waste removal, and it limits cell size.
So it's like trying to cram too much stuff into a small backpack.
Good analogy. And to overcome this challenge, cells have evolved different strategies to increase their surface area without drastically increasing their volume. For example, some cells have long, thin extensions, like nerve cells, which transmit signals over long distances. Others have finger-like projections called microvilli, like the cells lining our intestines, which increase their surface area for absorbing nutrients.
It's amazing how cell structure is so finely tuned to function. Okay, so now that we have a basic understanding of the two main types of cells, are we ready to delve deeper into the eukaryotic cell?
Absolutely. Let's take a closer look at the eukaryotic cell's inner workings and explore those fascinating organelles that make life possible.
Okay, buckle up. We're about to embark on a guided tour of the eukaryotic cell, a bustling metropolis of organelles, each with its own unique role to play.
Welcome back to our deep dive into the amazing world of eukaryotic cells.
I'm ready to explore more of this microscopic metropolis. Last time, we left off just as we were about to take a closer look at the eukaryotic cell's organelles. Where should we start our tour?
Well, you can't talk about the eukaryotic cell without mentioning its defining feature, the nucleus.
Yes, the cell's control center. That's where the DNA is stored, right?
Exactly. The nucleus is like a highly secure vault, protecting the cell's precious genetic information, the blueprints for, building and running the entire cell. It's enclosed by a double membrane called the nuclear envelope, and it's dotted with pores that act like checkpoints, regulating what goes in and out. Imagine it as a border crossing with, really strict security measures.
Okay, so it's not just a passive container. It actively controls access to the DNA, what's happening inside the nucleus.
Inside, you'll find the DNA organized into chromosomes, those thread-like structures that carry genes. The DNA is also associated with proteins, forming a complex called chromatin. And there's a distinct region called the nucleolus, where ribosomes are assembled.
Well, ribosomes, those are the protein factories of the cell, right? They translate the genetic instructions from the DNA into proteins. But I thought ribosomes were located outside the nucleus, in the cytoplasm.
You're right. Ribosomes are synthesized in the nucleolus, but then exported to the cytoplasm, where they actually carry out protein synthesis. Some ribosomes float freely in the cytoplasm, while others are attached to the endoplasmic reticulum, or ER.
I remember you mentioned the ER earlier. It's a network of interconnected membranes, kind of like a maze, right?
That's a good way to visualize it. The ER is this vast network of interconnected sacs and tubules that extends throughout the cytoplasm. And there are two types of ER, smooth ER and rough ER. The rough ER is studded with ribosomes, which gives it that rough appearance, and is involved in protein synthesis, folding, and modification.
Okay, so the rough ER is like a protein production and processing plant.
Exactly. It's like a workshop where proteins are assembled, inspected for quality control, and packaged for delivery.
What about the smooth ER? What's its role?
Well, the smooth ER lacks ribosomes and has a smoother appearance under the microscope. It's involved in a variety of metabolic processes, including lipid synthesis, carbohydrate metabolism, and detoxification of drugs and poisons.
So the smooth ER is like the cell's detox center and lipid production facility.
You got it. It's also involved in calcium storage, which is important for things like cell signaling and muscle contraction.
Wow, the ER is a multitasker. So what happens to the proteins that are synthesized and processed in the ER? Where do they go next?
Well, many of them are transported to the Golgi apparatus, another key organelle in the eukaryotic cell. Think of the Golgi as the cell's shipping and receiving center.
Okay, I'm picturing a busy sorting facility with packages coming in and going out in all directions.
That's a great analogy. The Golgi apparatus consists of these stacks of flattened membranous sacs called cisternae. Proteins and lipids from the ER arrive at the Golgi's receiving side, or cis-face, and then move through the Golgi, undergoing further modifications and sorting.
So like a series of checkpoints where the proteins get tagged and packaged for their final destinations?
Precisely. The Golgi adds molecular tags like carbohydrates to proteins, acting as addresses for delivery throughout the cell or even for export outside the cell. Finally, the processed proteins and lipids are packaged into vesicles, those tiny membrane-bound sacs, and shipped out from the Golgi's shipping side or transface.
It's amazing, this logistical feat happening on a microscopic scale. But what about cellular waste and recycling? Cells must have a way to get rid of old or damaged components.
That's where lysosomes come in. These membrane-bound organelles act as the cell's recycling and waste disposal system.
Okay, so like the cell's garbage disposals or recycling centers.
Exactly. Lysosomes contain powerful digestive enzymes that can break down macromolecules like proteins, lipids, carbohydrates, and nucleic acids into smaller components that can be reused or eliminated. They can even digest entire worn-out organelles or engulfed bacteria and viruses.
So they're like the cell's demolition crew and sanitation workers all in one, keeping things clean and tidy.
Exactly. And in plants and fungi, vacuoles play a similar role, although they're often much larger than lysosomes and have additional functions like storing water, ions, and pigments.
Oh, right. I remember learning about those large central vacuoles in plant cells. They can take up so much space. So we've covered protein synthesis, processing, and recycling. But all of these processes require energy. Where does the cell get its fuel?
That's a great question. And it brings us to two of the most fascinating organelles in the eukaryotic cell, mitochondria and chloroplasts.
Okay, let's dive into the world of cellular energy. First up, mitochondria, the powerhouses of the cell. I've always been intrigued by these organelles.
Mitochondria are remarkable. They're found in almost all eukaryotic cells and are responsible for cellular respiration, the process that converts the chemical energy stored in food molecules, like sugars and fats, into a form of energy that the cell can use, called ATP.
So they're like tiny power plants, generating the fuel that powers all of the cell's activities.
Exactly, and their structure is perfectly suited for this function. Mitochondria have not one, but two membranes, an outer membrane and a highly folded inner membrane. Those folds, called cristae, increase the surface area of the inner membrane, creating more space for the enzymes involved in ATP production.
It's like packing more power into a smaller space, maximizing efficiency.
Precisely. And here's a mind-blowing fact. Inside the mitochondria, within the matrix, you'll find its own set of ribosomes and even its own DNA.
Wait, mitochondria have their own DNA? That's incredible. What's the significance of that?
Well, that's a clue to their fascinating evolutionary history. It supports the endosymbiont theory, which proposes that mitochondria and chloroplasts were once free-living prokaryotic cells that were engulfed by early eukaryotic cells.
Whoa, it's like a cellular merger, but one where the smaller company retains some of its independence.
That's a great way to think about it. Over time, this relationship became mutually beneficial, with the engulfed prokaryotes evolving into the organelles we know today as mitochondria and chloroplasts.
That's an amazing example of symbiosis, where two organisms live together in a mutually beneficial relationship. Okay, so what about chloroplasts, the other energy-converting organelles? Those are found in plants and algae, right?
You're exactly right. Chloroplasts are responsible for photosynthesis, the process that captures light energy from the sun and uses it to convert carbon dioxide and water into sugars, which then serve as the primary source of energy for most ecosystems on Earth.
So they're like tiny solar panels, harnessing the sun's energy to create food.
An excellent analogy. And like mitochondria, chloroplasts also have a double membrane and their own DNA and ribosomes, which supports that endosymbiotic origin.
This is all so cool. We've seen how the nucleus controls the cell's activities, how the endoplasmic reticulum and Golgi apparatus handle protein production and processing, how lysosomes act as the cell's cleanup crew, and how mitochondria and chloroplasts provide the cell with energy. But there's still one piece of the puzzle missing. How do all these organelles stay organized and move around within the cell?
That's where the cytoskeleton comes in. It's much more than just static scaffolding.
Okay, I'm all ears. Tell me more about this dynamic network of protein fibers.
The cytoskeleton is this complex network of protein filaments that extends throughout the cytoplasm. It provides structural support, facilitates movement, and plays a role in cell signaling. Imagine it as a cellular highway system, transportation network, and communication hub all rolled into one.
That's a lot of responsibility for a bunch of protein fibers.
It is. And the cytoskeleton is made up of three main types of fibers: macrotubules, microfilaments, and intermediate filaments.
Okay, let's break those down. What are microtubules?
Microtubules are the thickest of the three. They're these hollow tubes made of protein subunits called tubulin. They play a crucial role in maintaining cell shape, guiding the movement of organelles, separating chromosomes during cell division, and forming structures like cilia and flagella, which are involved in cell movement.
Like cilia and flagella, those whip-like structures that propel some cells through their environment.
Precisely. Cilia are short, hair-like projections that often cover the entire surface of a cell, while flagella are longer, whip-like structures that are usually limited to one or a few per cell. Both cilia and flagella are composed of microtubules arranged in a specific pattern, and they're powered by motor proteins that slide along the microtubules, generating movement.
I've seen videos of those tiny creatures swimming around with their flagella. It's amazing how those tiny structures can generate so much movement.
It is remarkable. And speaking of motor proteins, they play a crucial role in transporting cargo along microtubules within the cell. Imagine them as tiny molecular couriers, carrying vesicles filled with proteins or other molecules to their destinations.
It's like a bustling highway system with trucks carrying goods all over the city.
That's a great analogy. Now let's talk about microfilaments. They're also known as actin filaments and are the thinnest of the cytoskeletal fibers. They're composed of protein subunits called actin and are involved in cell movement, muscle contraction, and maintaining cell shape.
Muscle contraction. So our muscles are powered by these tiny actin filaments.
That's right. Microfilaments interact with motor proteins called myosin to generate the force needed for muscle contraction. They also play a role in amoeboid movement, which is how some cells, like amoebas and certain white blood cells, crawl along surfaces.
It's like a microscopic tug-of-war.
Exactly. And finally, we have intermediate filaments, which are fibers with a diameter in between that of microtubules and microfilaments. They provide structural support and are involved in anchoring organelles in place.
So they're like the cell's internal scaffolding, keeping everything organized.
Precisely. They're also involved in forming the nuclear lamina, a network of protein filaments that supports the nuclear envelope.
Wow, we've covered so much ground. From the nucleus to the cytoskeleton, it's amazing how all these different components work together to create a functioning cell. But before we move on, I'm curious about what lies beyond the plasma membrane, the cell's outer boundary.
That's a great question. And it leads us to the fascinating world of cell walls and the extracellular matrix.
Okay, I'm ready to explore what lies beyond the cell's borders.
Well, in plants, the most prominent extracellular structure is the cell wall, a rigid layer that surrounds the plasma membrane, providing support, protection, and helping to regulate water uptake. Think of it as a suit of armor for the plant cell.
Yeah, I can see how that would be important for plants, especially for providing structural support. What is the cell wall made of?
The plant cell wall is primarily composed of cellulose, a strong and durable polysaccharide. It's like a tough, fibrous network that gives the plant cell its shape and rigidity.
So it's like a cellular exoskeleton. But what about animal cells? They don't have cell walls, do they?
No, they don't. Instead, animal cells have this complex network of molecules called the extracellular matrix, or ECM, that surrounds them. It's kind of like a cellular support system, but it's much more dynamic than that.
Interesting. What is the ECM made of and what does it do?
The ECM is composed of a variety of proteins and carbohydrates that are secreted by the cells. Collagen, which is a fibrous protein, is a major component, providing structural support. Other proteins, like fibronectin and laminin, help cells attach to the ECM and play roles in cell signaling and communication.
So the ECM is like a cellular social network, connecting cells to each other and their environment.
That's a great way to put it. The ECM is a dynamic structure that plays a crucial role in cell behavior, influencing everything from cell migration to tissue development.
This is all fascinating. I'm starting to see just how complex a single cell really is. I never realized how much was going on beneath the surface.
And we've only just scratched the surface. There's still so much more to discover about the intricate world of cells. But before we wrap up this part of our deep dive, let's touch upon one more important aspect of cellular interactions: cell junctions.
Okay, what are cell junctions and why are they important?
Cell junctions are specialized structures that allow cells to connect and communicate with each other. They're like cellular bridges or communication channels that facilitate interactions between neighboring cells.
I can see how that would be important for cells to function as a coordinated unit, especially in tissues and organs. Could you give me some examples of cell junctions?
Sure. In plants, these connections are called plasmodemata. They're channels that directly connect the cytoplasm of adjacent cells, allowing for the free flow of water, small molecules, and even some larger molecules like proteins and RNA between cells. This enables communication and coordination.
So it's like a shared cellular resource network. What about animal cells? What kind of cell junctions do they have?
Animal cells have a variety of cell junctions, each with its own specialized function. Tight junctions create a watertight seal between cells, preventing leakage of fluids. Desmosomes act like rivets, anchoring cells together into strong sheets, providing structural integrity. And gap junctions are channels that allow for the passage of small molecules and ions between adjacent cells, enabling direct communication and coordination.
Wow, it's like a whole city with different types of infrastructure connecting the buildings and allowing for communication and transportation. This deep dive has really opened my eyes to the incredible complexity and dynamism of cells.
It truly is a remarkable world waiting to be explored. And in the final part of our deep dive, we'll delve even deeper into some of the fascinating processes that make life possible.
Welcome back to our cellular adventure. We've journeyed through this bustling eukaryotic cell, explored its intricate compartments, and marveled at its dynamic processes.
We've seen how the nucleus governs the cell's activities, how the endoplasmic reticulum and Golgi apparatus work together to make and process proteins, how lysosomes act as the cell's cleanup crew, and how the cytoskeleton provides support and facilitates movement.
It's amazing how all these different components work together so seamlessly. But before we wrap up our deep dive, I want to revisit those fascinating organelles we talked about earlier, mitochondria and chloroplasts.
Ah, yes, the powerhouses and the solar panels of the cell. Their story is like a biological thriller, full of intrigue and mystery.
You mentioned the endosymbiont theory, which proposes that these organelles were once free-living prokaryotes. It's such a mind-boggling concept. What's the evidence for this theory?
Well, one really compelling piece of evidence is that both mitochondria and chloroplasts have their own DNA, separate from the DNA found in the cell's nucleus. And this DNA is circular, just like the DNA found in bacteria.
Oh, wow, that's fascinating. So it's like they have their own little instruction manuals independent of the cell's main blueprint.
Exactly. And not only do they have their own DNA, but they also have their own ribosomes, the protein-making machines of the cell. And these ribosomes are more similar in structure to bacterial ribosomes than to the ribosomes found in the cytoplasm of eukaryotic cells.
So they have their own protein-making equipment, like their own mini factories within the cell.
That's a great analogy. And to add to the evidence, both mitochondria and chloroplasts can replicate independently of the cell's normal division cycle. They divide on their own, much like bacteria do.
It's almost like they're living a double life. It's incredible to think about how these once independent organisms became integrated into the eukaryotic cell.
It is. And this integration has been incredibly successful, enabling the evolution of complex multicellular organisms like plants and animals.
Talk about a win-win situation. So to recap, mitochondria and chloroplasts have their own DNA, their own ribosomes, and they can replicate independently. All of this points to their origin as free-living prokaryotes. That's some pretty convincing evidence.
It is. The endosymbiont theory has really revolutionized our understanding of the evolution of eukaryotic cells.
This deep dive has been an incredible journey. We've gone from peering through microscopes to exploring the intricate world of organelles and delving into the evolutionary history of cells.
We've seen how cells are like tiny cities with specialized compartments, intricate transportation networks, and constant communication.
It's truly mind-blowing to think about all the complex processes happening inside each and every one of our cells, from building proteins to generating energy to communicating with their neighbors.
And we've only just scratched the surface. There's still so much more to discover about these fascinating units of life.
Well, on that note, we'll have to wrap up our cellular exploration for now. A huge thank you to our listeners for joining us on this incredible journey through the fascinating world of cells. We hope you've gained a newfound appreciation for the tiny universes that make life possible.
And remember, everything you learn in biology, from the behavior of organisms to the complexity of ecosystems, is ultimately rooted in this remarkable world of cells.
Until next time, keep exploring, keep questioning, and keep diving deep into the wonders of science.